In our last lecture, we have seen introduction to HIV/AIDS and structure of HIV virus. Let us now understand mechanism of HIV infection step by step. Welcome to SK Biology. The first step is HIV entry. HIV enters the human body through infected body fluids like blood, semen. The most common modes of transmission are unprotected sexual contact or transfusion of infected blood, as well as sharing contaminated needles and mother to child transmission. After entering the body, the second step is infection of macrophages. HIV primarily infects macrophages, one of the body's most important immune cell. The virus binds to receptor on the macrophage surface and enters the cell and releases its RNA along with viral enzyme. The important neat point is HIV is not transmitted through mosquito bites, hugging, handshakes, sharing food, tears, or sweat. And macrophages are the first cells infected by HIV and acting as a HIV factory. The next step is reverse transcriptase action. Inside the macrophage, the viral enzyme reverse transcriptase converts the viral single-stranded RNA into double-stranded DNA. I repeat, it converts single-stranded RNA into double-stranded DNA. This process is called as reverse transcription. Neat point, reverse transcriptase is the characteristics enzyme of retroviruses. Next step is formation of viral DNA. With the help of reverse transcriptase enzyme, the RNA of virus is converted into viral DNA. This newly formed viral DNA is called as proviral DNA. It is now ready to become a part of the host cell's genetic material. Neat point, HIV carries an RNA genome, but after reverse transcription, it forms DNA inside the host cell. Next step is integration into host DNA. The viral DNA enters the nucleus and integrates with the DNA of macrophage with the help of enzyme integrase. Once integrated, it becomes a permanent part of host genome and uses the host cell machinery to produce viral components. Neat point, integrated viral DNA is called as provirus. Next and most important step in macrophages is virus production. The infected macrophages synthesizes viral RNA and proteins, assemble new HIV particles and releases them. These newly formed viruses go on to infect more immune cells. Neat point, macrophages can survive for a long time while continuously releasing new HIV particles, and because of this nature, macrophages are called as factory of HIV virus. Next step in HIV infection is infection and decrease of CD4 T cells. Here, the newly produced HIV particles from macrophages infects CD4 plus helper T lymphocytes, which are the main coordinators of immune response. After infection to CD4 T helper cells, HIV multiplies, destroying these cells. To enter inside CD4 T cells, HIV uses CD4 receptors. Initially, healthy CD4 count is higher than that of infected CD4 T cells. Next step in CD4 T cell is, as HIV multiplies, it gradually destroys helper T cells. The number of CD4 cells decrease progressively, weakening both humoral as well as cell-mediated immunity. So, this step is immune dysfunction, which results into impaired activation of B lymphocyte, cytotoxic T cells as well as macrophages. With less number of helper T cells, immune system becomes unable to fight infections effectively, which leads to high susceptibility to opportunistic infections like pneumocystis pathogen, cancer, candida, etc. All this immune dysfunction lastly end up in the development of AIDS. When immune function is severely compromised because of marked decline in CD4 cells, the individual develops acquired immunodeficiency syndrome. At this stage, recurrent infections, certain cancers, and severe weight loss are common. Important need points. CD4 plus helper T cells are the primary target cells of HIV. The CD4 count is an important indicator of disease progression. AIDS patients usually die due to opportunistic infections, not because of HIV directly. This is all about mechanism of HIV infection.